Electrophoresis types and clinical applications. So, types of electrophoresis. Electrophoresis techniques, they are classified based on types of sample, that is DNA, RNA and protein, and support medium like gel, paper or capillary electrophoresis, and the principle of separation, be it charge, size or isoelectric point. So starting off with agarose gel electrophoresis, it is used for DNA and RNA. Medium is agarose. Agarose is a polysaccharide that is derived from a seaweed, and it separates nucleic acids based on size. DNA is negatively charged, move towards the NO, which is positively charged. Smaller fragments, they migrate faster. Visualization is done with stains like ethidium bromide or cybergreen under UV light. Application is PCR product analysis, DNA fingerprinting and restriction fragment analysis. Second is polyacrylamide gel electrophoresis, that is also abbreviated as PAGE. It is used for proteins and small nucleic acids. Medium is polyacrylamide. It separates molecules by size but also is affected by charge and shape. It provides high resolution and is suitable for small molecules. Application is protein profiling and enzyme analysis. Next comes is SDS-PAGE. SDS stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is typically used for proteins. Medium is polyacrylamide in addition to SDS, that is sodium dodecyl sulfate, which is a detergent basically. So SDS denatures the proteins and coats them with a uniform negative charge, so that separation is based only on size. Small proteins, they migrate faster. Application is at molecular weight determination of proteins, purity check and Western blotting. Next is isoelectric focusing. Isoelectric focusing is used for proteins. Medium is polyacrylamide gel with pH gradient. Here, the proteins migrate until they reach a pH equal to the isoelectric point. At isoelectric point, the net charge is 0. Migration stops. Isoelectric point is basically a pH at which the protein carries no net charge, right? So the application here, it is used in the precise separation of proteins with similar molecular weights but different isoelectric points. It is a part of 2D electrophoresis. 2D electrophoresis, that is 2-dimension gel electrophoresis, it is also called as 2D PAGE. It is used for complex protein mixtures. So the steps is that in first dimension, there is the separation by isoelectric focusing, that is separation on the basis of isoelectric point, and then the second dimension takes place, that is the separation by SDS-PAGE, which separates the molecules, which separates the particles based on their size. So the application is proteomics, biomarker discovery, disease-related protein studies. Next comes capillary electrophoresis. Here it is used for small ions, nucleic acids and peptides. Medium is buffer-filled narrow capillary tube. It is very fast and high resolution, and separation here is based on charge to size ratio. Application is in clinical diagnostics, DNA sequencing and drug analysis. Next comes paper electrophoresis. It is used for amino acids and small peptides. Medium is the filter paper, and it is one of the oldest techniques. Limited resolution, but it's very simple. Application is educational purposes, historical use in amino acid separation. Then comes cellulose acetate electrophoresis. It is used for hemoglobin variants, serum proteins. Medium is cellulose acetate strips. It is simple and rapid. It is widely used for HP electrophoresis to detect sickle cell disease and thalassemia. This is the summary table. Thank you.